Hi guys, so today we're going to be talking about plant transpiration and sensitivity. This will include how water moves through plants, the factors that affect the rate of transpiration, transpiration being the movement of water through plants. Tropisms, that's how plants grow. Remember, plants can't move around their environment. Most plants can't move around their environment. So instead what they do is they grow towards or away from different stimuli. And linked to this, we're going to be talking about plant hormones. So to start off with, we're looking at plant transport. Now, plants need a very different form of transport mechanism than we do, right? We have the circulatory system, which is needed to move nutrients and oxygen and stuff around our bodies at speed. But plants can do things much slower because their rate of metabolism, that is the rate of chemical reactions in their body, is much slower. Plant cells have a number of different requirements. They need oxygen for respiration. Obviously, through the night, they're not producing oxygen, and they need to get that. So stomata are these small openings on leaves that can open up and allow gas exchange. They need organic nutrients, such as sugars, which they make using photosynthesis. They need carbon dioxide for photosynthesis. Again, gas exchange happens through stomata, these little holes, these little pores in the opening of their leaves. And they need water and inorganic ions, which they take up from their roots. Plant transport systems then very different from ours. They have two main transport networks. There's the xylem and the phloem. Xylem vessels are all about carrying water, and they carry water up from the roots up through the plant. Phloem tissues move the products of photosynthesis in both directions, because obviously a lot of the nutrients from photosynthesis will need to move down to the roots to supply those cells with energy, which they aren't obviously able to photosynthesize themselves, and they also need to provide energy to photosynthesis. Products need to be moved, say, to fruit or seeds or other things like that. So xylem vessels moving water, this process is transpiration. Phloem vessels, the process is called translocation. We're not going to look much at the movement of translocation. We're going to instead focus on transpiration, much more likely to be assessed. So transpiration starts off with water in the soil being transferred through root hair cells into roots, then up xylem vessels through the stem to the leaves, and then out into the atmosphere. By far the majority of water that a plant takes up goes all the way through the plant and out into the atmosphere. Only a small amount will be used in photosynthesis. Soil has a very high water potential compared to the atmosphere. And each of these things, in turn, has a lower water potential than the one before it. So we get movement of water from the soil to the atmosphere. This starts off with root hair cells of the roots. These are cells that have these long hairs coming off them, massively increasing their surface area. This enables the plant to take up as much water as possible by osmosis. In order to do this, the plant will pump mineral ions by active transport into their root cells. The water will then follow by osmosis and so move into the plant. The water then moves up into the xylem vessels via these two pathways, the symplast and apoplast pathway. Maybe you can spot on the diagram how they're slightly different. But essentially what it does is it moves into the middle of the stem and starts its journey up the xylem vessels. Xylem vessels are hollow tubes. They're cells that have carried out apoptosis, that is programmed cell death, killed themselves to create these long continuous tubes running from the roots all the way up to the leaves. In these tubes, water molecules are moved using cohesion and adhesion, two critical properties of water. Cohesion, remember, is between water molecules. Adhesion is when they're attracted to other things. And in the case of xylem vessels, xylem has got this polysaccharide called lignin, which is very, very strong. It's like supercellulose, that prevents the tubes from collapsing in on themselves. Imagine trying to suck water through a paper straw, like one of those ones you might get at a fast food restaurant. The outer covering, it might just collapse. So in order to keep this strong, to stop it sucking in on itself, the xylem vessels have a lot of lignin in their walls. So the water molecules are pulled up in a continuous chain all the way up the xylem vessels, with cohesion between each one and adhesion between them and the lignin around them, enabling to pull them up. It's important to mention at this point that, of course, these xylem vessels are a crucial adaptation of plants when it comes to evolving to live on land. The xylem vessels have two functions. One, they're enabling water to be transported to all the parts of the plant, which, if the plant was living underwater, would not be an issue. The second key adaptation to life on land here is that the lignin in the xylem vessel walls gives them a lot of strength. When a plant is living in water, the water around it provides a lot of support. But when it's out in the air, think about seaweed. If you take it out of the sea, it just flops over. It's not very strong. The lignin provides that strength for land terrestrial plants in order that they can be tall and compete for sunlight. So these are two critical adaptations for life out of water. Root systems would be another one, and the production of seeds and seed cases to prevent the gametes from drying out would be another. Okay, one more thing I should mention, of course, fruit and wind-blown seeds would be other adaptations too for life on land. So the water moves up the xylem vessel, goes all the way up to the leaf. This is a cross-section of a leaf here. And you can see the water coming in on this orange line here is moving from cell to cell by osmosis. As I said, most of the water will then evaporate into these air spaces and diffuse out of the stomata. The stomata are these small holes, these openings in the base of most leaves. Now, it's important to understand that this is not ideal for the plant. The plant would ideally keep as much water as possible, but they have to lose some because they're going to need to do gas exchange. When you're talking about transpiration, think it's evaporation that's happening from the leaf. What factors will affect it? So there's a water potential gradient between the cells and the atmosphere, and so water will be lost from the stomata. This is transpiration, this flow of water through the plant and out by evaporation. Now, there's a lot of environmental factors that can increase, or if they're absent, decrease the rate of transpiration. If it's warm or hot, then evaporation will happen faster. If it's windy, because the molecules have got more energy, if it's windy, the moist air that evaporates from the leaf will be carried away in the wind, thereby maintaining a steep concentration gradient of water potential between the leaf and the air around it. If the air is very dry, that will also mean that there's a steep concentration gradient, so diffusion will happen quickly. Obviously, the opposite of these are also true. If it's cold, transpiration will be slower. If the air is still, then transpiration will be slower. And if it's very humid, transpiration will be slow. In fact, at 100% humidity in the air, transpiration stops because the water will not be evaporated. So normally in these scenarios that are on the slide, there is a high water potential gradient between the environment and the spongy mesophyll. That's the cells around these air spaces. But if the water potential gradient is lowered, because there's less of a difference between the water potential of the cells and the surrounding air, then transpiration will slow down because evaporation will slow down. Now, many plants have got their stomata on the underneath surface of their leaf. This is because it is cooler underneath of the leaf, so less evaporation will happen and the plants will lose less water. Remember, these stomata are not there so that the plants can lose water. The plants don't want to lose water, even if there's often quite a lot available. Instead, the reason that they're there is so that gas exchange can happen. Carbon dioxide and oxygen can pass through and respiration and photosynthesis can occur. So that's why they're normally underneath of the leaf. The cells on either side of a stomata are called the guard cells. And these guard cells are very special because they can open and close. If you look at the picture in the bottom right here, this little animation, you can see that as the guard cells fill with water, they move apart from one another. As they swell, they actually open the stomata. Now think about this. This is clever for a number of reasons. One, if the plant was very dehydrated, it needs to keep water. And if the guard cells are dehydrated, well, they're going to be shriveled and so closed. When they swell up with water, they open up. So the plant has enough water, it can do gas exchange. Not only that, but plants want to keep as much water as possible when they don't need it, when they don't need gas exchange. So they keep the water when they don't need gas exchange, and the way that they do this is really clever. They close their stomata at night. Some will even close it during the hottest part of the day to retain water. But a lot of plants will close their stomata at night. And then, amazingly, the first thing that happens is at dawn they get blue light receptors that are detected by receptors on the guard cell wall. So these receptors, they detect blue light. The receptor signals to H+ ATPases on the guard cell's plasma membrane to start pumping hydrogen ions out of the guard cell. That creates a more negative charge within the guard cells. And so potassium ions will flow into the guard cell. H+ pumped out, potassium flows in. When the potassium flows in, then of course we get osmosis occurring. Because the osmotic pressure of the guard cells is lower, water will follow by osmosis. Water flows into the guard cells, and the stomata will open. Okay. Really, really clever little mechanism. So actually, plants detect the light that's present, the blue light at dawn, and open their stomata, ready for gas exchange, ready for photosynthesis. One way we can measure the rate of transpiration is with this piece of apparatus. This is a very useful piece of apparatus to know about. It's called a potometer. Potometers effectively just measure the rate of uptake of water by a plant. There is another way of doing this you can do, which is if you wrap say a root ball in foil, you can prevent evaporation there, and change in mass can also tell you how much water a plant has taken up. But this is a way that you might need to know as well. This is called a potometer, and you can see that as water passes through the plant, the water is taken in, and we can read that off the scale. So for example, you could put it under different lighting conditions, with a fan, under different temperature conditions, under different humidities. Some plants, called xerophytes, such as marram grass, have special adaptations to reduce water loss even more. These xerophytes are plants adapted to very low water conditions. Marram grass lives on sand dunes, where there's very little water because the water easily flows through the sand. The marram grass rolls its leaf up. The stomata are very sunken, and there are leaf hairs on the surface of the leaf. Rolling the leaf up causes this air inside to be trapped, keeping a small concentration gradient between the stomata and the cells here, and this air in the space. In other words, it's trapping this layer of moist air. The leaf hairs help with that, and they prevent air movement, so the moist area is not carried away from the grass. So we have a lower concentration gradient of water, and so the water stays in the plant. Having the sunken stomata, having them really deep down inside the plant as well, prevents evaporation from happening quickly. Finally, plants, and this is the case for a lot of plants that live in very dry conditions, have a very waxy layer on their outside. This is like a wax rain jacket, except instead of trying to keep water out, it's keeping water in. So water cannot pass through the waxy cuticle. All of these are structures that lower the rate of transpiration, but enable gas exchange to still take place. So this is a picture of one of those structures. You can see these hairs coming off the leaf here, preventing air movement, and the stomata would be right down in the base of these pits. Next up, I want to talk about plant hormones. So plant tropism is a growth response caused by hormones that cause plants to either grow away from or towards a stimulus. Now, you've probably seen this in plants growing towards light. If you leave them in a window, they'll bend in that direction. That's called phototropism. They also are able to detect gravity. Shoots grow up and roots grow down, gravitropism. Thigmotropism is a response to touch. You may have seen vines, for example, that curl themselves around supporting branches in order to climb up other trees. There are positive and negative tropisms. Positive means you move towards the stimulus, negative is away from it. And different parts of a plant can respond in different ways. A coleoptile is the young shoot of a seedling, and if we take, say, an oat seedling here, this is where the growth of a shoot occurs, right at the tip. So this is the region of cell division, and then right behind it we have a region of elongation. The cells divide and then they stretch. So when growth happens, it happens like so. Now, there's some cool experiments you can do around this. If you chop the tip, if you leave it as normal, it grows. But if you chop the tip off, the base does not grow. Interesting. Therefore, there's something in the tip that's enabling the zone of elongation to stretch. If we cut it off and then replace it, the shoot grows. So it's not an attachment issue. Something is passing from the tip into the shoot. In other words, we need the tip for the growth of the shoot. Now, here's a really cool experiment. You take an agar block. Chemicals can diffuse through this, but there's no physical connection. When we do that, elongation still occurs in the zone of elongation, and the shoot grows. In other words, there is a chemical that is being produced at the tip, which is diffusing through the agar into the zone of elongation, promoting its growth. Next up, we can take the tip and place it onto the agar block separately. When we slide it across onto the top of the shoot, amazingly, it still grows. So this tells us that the chemical is diffusing into the block, then it's diffusing into the zone of elongation, stimulating the plant to grow. Here's another cool thing you can do. You can chop the tips off the coleoptiles and place it on either side of the zone of elongation. That causes the shoot to grow in different directions, depending on which side you place it on. Whichever side it's on, that side grows more, and so the plant bends. The reason for this is the chemical is diffusing down vertically into the zone of elongation adjacent to it. So what is it that's doing this? Auxins. Auxins are produced by the tip of the shoot, and they exert their effects on the zone of elongation. They stimulate cell division and elongation. Now, interestingly, there's a different response for roots and shoots. In roots, the auxins low concentrations stimulate growth, and high auxin concentrations actually inhibit growth. So it may seem strange. In shoots, it's different. Low auxin concentrations do not stimulate growth, and high auxin concentrations do. Now, the reason that that is critical is because roots and shoots need to respond in very different ways to the same stimulus. So light, for example, causes an uneven distribution of auxins in the region of elongation of the shoot. Notice, all the auxins have moved away from the light. There's a higher concentration of auxins in the shaded side of the root, and so when it grows, the shoot will bend towards the light. You may have seen this with tomato plants bending towards the light in your garden or on a windowsill. So that side will grow faster. It's a clever way for the plant to orientate itself towards the light. Roots behave in exactly the opposite manner. They will bend away from the light. Think about the advantage of that. They're going to bend themselves down towards the soil. Geotropism, or gravitropism, is linked to gravity. Okay. The effect of gravity also causes uneven distribution of auxins. Auxin concentration on the lower side of the root is higher than the upper side. Now, obviously in a shoot, that would make it bend upwards. But in a root, because auxins inhibit growth, they will actually bend downwards towards going into the ground. Because of this difference in response between roots and shoots, they behave in opposite ways, and so they move in the direction that they need to for the plant to remain healthy. So auxins stimulate cell elongation. They actually cause proton pumps to activate and lowering the pH around the cell walls. And I think this is really cool. It lowers the pH, which basically weakens the cell wall. With the increased turgor pressure, that causes the cell wall to expand and elongate, allowing the cell to grow. We can actually use auxins to kill certain plants by providing them in really high concentrations. The plant will grow faster than it has the energy to support and will run out of its energy stores. That's how we kill various weeds. There are other plant hormones too. Cytokinins, which stimulate cell division. A proper ratio of auxins and cytokinins will enable good cell growth. Gibberellins work with auxins to stimulate elongation. They are part of this loosening cell wall process. If you're going to remember any of these, please remember auxins, but these are quite useful ones too. Gibberellins are important in ceasing dormancy of seeds, right? Seeds need to stay dormant for a long time, but then they can be released from their dormancy. Abscisic acid is a really cool one. Abscisic acid slows growth. It prevents it prevents seeds from germinating too early. So for example, tomatoes contain a very high concentration of abscisic acid to prevent the seeds inside from germinating when they're still on the plant. It's not until the plant has maybe been eaten by a human, pooped, and released into the environment free from abscisic acid that it will start to grow. This ensures that the fruit is carried a long way from the original parent plant, which is what the original parent wants, because it doesn't want to compete with its offspring. Ethylene is another plant hormone. It's a gas, and it's used to determine programmed cell death, apoptosis. This is how fruit gets ripe. Here's a really interesting picture. We can see, oops, oh no, I don't have it on here anymore. Anyway, if you expose bananas to ethylene gas, they go black really quickly. One bad fruit actually does trigger ripening in the other fruits. This is actually an evolutionary strategy. Enabling all the fruit to ripen at the same time means that you're much more likely to attract herbivores to spread the seeds. Finally, plants can use chemicals in other ways, and I just want to briefly touch on this because I think it's incredibly cool. For example, if a pest species is feeding on a plant, this will actually trigger a signal transduction pathway using the chemical in the pest's saliva. The plant will release volatile attractants that will recruit parasite wasps to lay their eggs within the caterpillars and kill the caterpillars. It's a very clever little signaling pathway to attract wasps and get rid of their own pests, the caterpillars. Very cool. Right, that's it for today. Enjoy the rest of your day.